Structure-activity relationships of phenylethanolamines. Part 2. Before you watch this video, make sure you have seen the previous video of SAR of Phenylethanolamines Part 1. It will help you understand this video better and avoid confusion. You can find the link to the previous video here or in the description box below. Along this video, we will use some abbreviations. SAR, for structure-activity relationships. And PEA, for phenylethanolamines. COMT, for catechol-O-methyltransferase enzyme. And MAO, for monoamine oxidase enzyme. Which both are metabolizing enzymes for noradrenaline. We knew that noradrenaline is synthesized and stored inside vesicles to be released when needed to exert their effects by binding to specific adrenergic receptors. There are two types of adrenergic receptors, alpha and beta receptors. Some receptors are postsynaptic receptors such as alpha-1, beta-1 and beta-2. Alpha-1 receptors found on vascular smooth muscle cause their contraction such as blood vessels and neck of stomach, urinary bladder, and prostate. In contrast, beta-2 relaxes the smooth muscles. They are predominantly present in airway smooth muscles causes bronchodilation. They also exist on uterine muscles causes their relaxation. Beta-1 receptors are predominantly found in three locations, the heart, the kidney, and the fat cells. Thus, activation of beta-1 receptors in heart increasing heart rate and contractility and increase renin secretion from kidney. On the other hand, alpha-2 are presynaptic receptors. Let us ask a simple question. The existence of postsynaptic receptors makes sense. However, what is the need for receptors that are located on presynaptic neurons? In fact, they act as noradrenaline release regulator. When activated, they reduce further release of noradrenaline and other neurotransmitters in both the central and peripheral nervous systems. They act as a negative feedback system. At this stage, it is time to go back to our topic. Let us illustrate the SAR of PEA. Basically, PEA can be divided into three parts: amine group, phenyl group and the ethyl bridge that connects them. Let us start with amino group. Amino group can be primary as noradrenaline, or secondary as in adrenaline. Based on fact that the binding site of beta receptors is wider than that of alpha receptors, therefore, beta can accommodate larger molecule than alpha. It seems that we found a selectivity clue. The clue is the R size. By increasing R size, the activity at alpha receptors decreases while activity at beta increases. If R is a small group such as hydrogen or methyl, the activity at both receptors is maximal. When R gets larger to be isopropyl, the activity shifts to beta receptors. R can also provide selectivity for different beta subtypes. Tertiary butyl affords selectivity for beta-2 receptors. By shifting to phenyl group, the nature, number and position of the substituents on the phenyl ring greatly matters. Generally speaking, the more polar and hydrophilic the substituent is, the less lipophilic and more water-soluble the PEA is. This means that it will have a harder time crossing the blood-brain barrier and reaching the CNS, where many of the neurotransmitter receptors are located. On the other hand, the more lipophilic and hydrophobic the substituent is, the more easily it can cross membranes and enter the CNS. Dopamine, noradrenaline and adrenaline are catechol derivatives, so they have two hydroxyl groups at position 3 and 4 on the benzene ring. Both hydroxyl groups are very important to bind to both alpha and beta receptors by hydrogen bonding. Actually, 3-hydroxy retains alpha-1 selectivity, while 4-hydroxy retains beta selectivity. Hence, catechol ring has optimum activity to both receptors, but at the same time is liable to metabolism by COMT enzyme. Therefore, catechol derivatives have poor oral activity due to their hydrophilicity and liability to break down by COMT. So, Any modification in position of hydroxy groups on benzene ring has a significant effect on both activity and selectivity. Furthermore, any modification at the 3-hydroxyl group, which is the site of COMT attack, this can provide resistance to the enzyme. If the 3-hydroxyl group is converted to hydroxymethyl, hydroxyl group is now no longer a phenolic. This provides beta-2 selectivity as well as COMT resistance and consequently, the oral activity. Also, if catechol ring converted to resorcinol, which is 3 and 5 dihydroxy derivative, also will provide beta-2 selectivity in COMT resistance. While 2 and 5 dihydroxy or dimethoxy provides alpha-1 selectivity. In case of that the two OH groups were removed, it means that the affinity to both receptors will obviously decrease in case of phenylethanolamines and is completely abolished in case of phenylethylamines. The absence of hydroxyl groups on benzene ring affords phenylethanolamines that active both directly on receptors with low affinity as well as indirectly by increasing the release of adrenergic neurotransmitters. While the absence of hydroxyl groups on benzene ring in phenylethylamines results in loss of efficacy that affords only indirect active compounds or may lead to antagonists. Final issue regarding phenyl group, the presence of lipophilic substituent on benzene ring such as methyl or chlorine instead of hydroxyl groups shifts the activity to alpha receptors. This will be discussed in more detail at a future opportunity. Next, let's look at the carbon bridge, which is 2-carbon chain between the phenyl ring and the amino group. Regarding to the bridge length, a 2-carbon bridge retains the activity, while increasing the length to 3 or 4 atom bridge provides alpha and beta antagonists. Most frequently, a hetero atom such as oxygen or nitrogen, is inserted in the bridge in case of antagonists. In case of the 2-carbon bridge, the carbon near amino group is alpha carbon while the other is beta. Alpha carbon is the target of MAO, so, methylation of this carbon affords MAO resistance as well as alpha-2 selectivity. Beta carbon carries hydroxyl group that converts dopamine into noradrenaline. It converts the phenylethylamines into phenylethanolamines. Its importance arises in case of absence of hydroxyl groups on benzene ring, because it keeps the chance of direct acting on receptors. Also, it alters the compound polarity and hence its ability to pass blood-brain barriers to exert their effect on CNS. Furthermore, the presence of hydroxyl group on beta carbon converts it into chiral carbon. Thus, generates two stereoisomers, R-noradrenaline and S-noradrenaline. The naturally biosynthesized and the most active is the R1, in which the hydroxyl group is oriented above the plane. Thank you for watching this video. I hope you enjoyed it. There's more to come. Stay tuned. See you in the next video.